Summary of origin and history of life. Event time period description. Formation of Earth. 4.6 billion years ago the Earth forms from cosmic dust and gas. Abiogenesis. 4.0 billion years ago life arises from non-living matter through chemical reactions. First evidence of life. 3.5 billion years ago microbial life forms, such as bacteria and archaea, appear. Photosynthesis and oxygenation. 3 billion years ago photosynthetic bacteria evolve, increasing atmospheric oxygen. Eukaryotic cells evolve. 1.8 billion years ago complex cells with. You said, continue ChatGPT said, ChatGPT. Nucleus and organelles, such as mitochondria and chloroplasts, evolve. The endosymbiotic theory suggests eukaryotes arose from symbiotic relationships between prokaryotes. First multicellular life. 1 billion years ago, simple multicellular organisms, likely composed of colonies of cells, appear. Cambrian explosion. 541 million years ago, rapid diversification of life forms, with the appearance of many major animal phyla. Rise of vertebrates. 500 million years ago, vertebrates, including early fish, evolve. Colonization of land. 450 million years ago, plants and arthropods begin to colonize land, followed by vertebrates. Mass extinctions throughout Earth's history. Several mass extinctions, e.g., Permian Triassic, Cretaceous Paleogene, shape evolutionary history. Evolution of mammals. 65 million years ago, mammals become the dominant land animals following the extinction of the dinosaurs. Human evolution. 2.5 million years ago to present, early human ancestors emerge, with modern Homo sapiens evolving around 200,000 years ago. Type. Gate practice questions and answers for Origin and history of life on Earth. 1. Gate 2018 Question 1. The appearance of oxygen in Earth's atmosphere, which led to the Great Oxidation Event, was due to the evolution of A. Photosynthetic bacteria. B. Eukaryotes. C. Atmospheric nitrogen. D. Volcanic eruptions. Answer. A. Photosynthetic bacteria. Explanation. The Great Oxidation Event, which occurred about 2.4 billion years ago, was caused by the evolution of photosynthetic bacteria, specifically cyanobacteria. These bacteria produced oxygen as a byproduct of photosynthesis, leading to the gradual accumulation of oxygen in the atmosphere. 2. Gate 2017 Question 2. Which of the following is a major feature of the endosymbiotic theory for the origin of eukaryotic cells? A. Eukaryotic cells were formed by the fusion of two prokaryotic cells. B. Prokaryotic cells engulfed other prokaryotic cells, which later evolved into mitochondria and chloroplasts. C. Eukaryotic cells are products of abiogenesis. D. All modern eukaryotic cells originated from a single primordial eukaryotic ancestor. Answer. B. Prokaryotic cells engulfed other prokaryotic cells, which later evolved into mitochondria and chloroplasts. Explanation. The endosymbiotic theory proposes that mitochondria and chloroplasts in eukaryotic cells are the result of a symbiotic relationship between a host cell and engulfed prokaryotes, which eventually became permanent components of the cell. This theory is supported by genetic evidence showing that mitochondria and chloroplasts have their own DNA, distinct from the nuclear DNA. 3. Gate 2015 Question 3. Which of the following geological periods is associated with the Cambrian explosion? A. Precambrian. B. Cambrian. C. Ordovician. D. Devonian. Answer. B. Cambrian. Explanation. The Cambrian explosion refers to a period around 541 million years ago during the Cambrian period, marked by the rapid diversification of life, including the appearance of many major animal phyla. It is considered one of the most significant events in the history of life on Earth. Key concepts recap. Abiogenesis, the theory that life originated from simple organic compounds through natural chemical processes. Endosymbiotic theory, suggests that eukaryotic cells evolved from symbiotic relationships between primitive prokaryotes and engulfed bacteria. Great Oxidation Event, the accumulation of oxygen in Earth's atmosphere due to the photosynthetic activity of cyanobacteria. Cambrian explosion, a period of rapid diversification of life during the Cambrian period, marking the appearance of major animal groups. Conclusion and application. Understanding the origin and history of life on Earth allows us to contextualize the evolution of living organisms and ecosystems. The history of life reflects the interplay between biology, geology, and chemistry, shaping the modern world. Evolutionary theories like abiogenesis, endosymbiotic theory, and the Great Oxidation Event highlight the dynamic processes that have shaped life on Earth. In preparation for GATE, it's crucial to understand the timelines and mechanisms that contributed to the rise of life in the Earth's biosphere. Recognizing the significance of major events such as the Cambrian explosion, the origin of photosynthesis, and the emergence of eukaryotic cells will help in answering questions related to the origin of life, evolutionary processes, and diversity. You said. Diversity and classification of life, ChatGPT said. ChatGPT. Diversity and classification of life. The diversity of life on Earth is vast, encompassing millions of different species across various habitats. To manage this complexity, scientists use classification systems that categorize organisms based on shared characteristics and evolutionary relationships. The study of this diversity and classification system is essential to understanding the fundamental principles of biology, evolution, and ecology. 1. Concept of biodiversity. Biodiversity refers to the variety and variability of life forms within a given ecosystem, region, or on Earth as a whole. It is generally studied on 3 levels. Genetic diversity, variability in the genetic makeup among individuals of a species. Species diversity, the variety of species within a given ecosystem or on Earth. Ecosystem diversity, the diversity of ecosystems in a particular region, including forests, wetlands, oceans, etc. Biodiversity is influenced by both evolutionary processes and ecological interactions, and it plays a critical role in maintaining ecosystem services, such as pollination, nutrient cycling, and climate regulation. 2. Principles of biological classification, taxonomy. Taxonomy is the branch of biology that deals with the classification of organisms. It involves organizing living organisms into hierarchical categories based on their shared characteristics. The main goal is to categorize organisms into groups that reflect their evolutionary relationships. The system of classification was pioneered by Carl Linnaeus in the 18th century, who developed a hierarchical system of naming and grouping organisms known as binomial nomenclature. This system assigns each organism a 2-part Latin name, one for the genus and another for the species. Taxonomic hierarchy. The taxonomic hierarchy is a nested system in which each level of classification is broader than the one below it. The major taxonomic ranks are as follows. Domain, the highest and broadest rank. All life forms are classified into 3 domains. Bacteria, single-celled organisms without a nucleus, prokaryotes. Archaea, single-celled organisms with distinct biochemistry, also prokaryotes. Eukarya, organisms with eukaryotic cells, including animals, plants, fungi, and protists. Kingdom, the second highest rank. Major kingdoms include Animalia, animals, plants, fungi, protista, protists. Monera, prokaryotes, now divided into bacteria and archaea. Phylum, a group of related classes of organisms. For example, Chordata is a phylum that includes all animals with a notochord, e.g., vertebrates. Class, a group within a phylum, such as Mammalia, which includes all mammals. Order, a group within a class. For example, Carnivora carnivores is an order within the class Mammalia. Family, a group within an order. For example, Felidae the cat family is a family within the order Carnivora. Genus, a group of closely related species. For example, the genus Panthera includes lions, tigers, leopards, and jaguars. Species, the most specific level of classification, representing a group of organisms capable of interbreeding and producing fertile offspring. For example, Panthera leo is the species name for the lion. Binomial nomenclature. Example, the scientific name of humans is Homo sapiens, where Homo is the genus and sapiens is the species. 3. Classification of life forms. The diversity of life on Earth is immense, and it can be categorized into several broad groups based on the characteristics of organisms. Below are the primary groups based on the taxonomic system. A. Prokaryotes, bacteria and archaea. Bacteria, single-celled organisms that lack a membrane-bound nucleus. They are found in virtually every environment on Earth. Archaea, similar to bacteria but have distinct biochemical and genetic features. They often live in extreme environments, such as hot springs and salt lakes. B. Eukaryotes, eukarya domain. Eukaryotes have cells with a nucleus and membrane-bound organelles. Eukaryotic organisms are classified into several kingdoms. Kingdom Animalia, animals. Multicellular, heterotrophic organisms, they consume organic material for energy. They exhibit complex nervous and muscular systems. Examples, humans, birds, fish, insects, etc. Kingdom Plantae, plants. Multicellular, autotrophic organisms, they produce their own food via photosynthesis. Examples, trees, flowers, grasses, and ferns. Kingdom Fungi. Primarily non-photosynthetic organisms that absorb nutrients from decaying organic matter. They play important ecological roles as decomposers. Examples, mushrooms, yeasts, molds. Kingdom Protista. A diverse group that includes unicellular organisms and simple multicellular organisms. Some are autotrophic, e.g., algae, and others are heterotrophic, e.g., protozoa. Examples, amoeba, paramecium, seaweeds. Kingdom Chromista. Includes algae and other simple organisms that are distinct from plants. Examples, brown algae, diatoms, water molds. 4. Methods of classification. Morphological classification. Organisms are classified based on physical characteristics such as size, shape, color, and structure. Genetic classification. This method uses molecular data, such as DNA, RNA, and protein sequences, to determine evolutionary relationships. This method is particularly important in modern taxonomy, as genetic evidence can uncover hidden relationships between organisms that may not be apparent from their morphology. Cladistics. A method of classification that groups organisms based on shared derived characteristics, synapomorphies. It focuses on evolutionary relationships rather than overall similarities. 5. Phylogeny and evolutionary trees. Phylogeny refers to the evolutionary history and relationships among organisms. Phylogenetic trees, also known as evolutionary trees, are diagrams used to depict these relationships. Cladograms, diagrams that show the evolutionary relationships based on shared characteristics. They are constructed using data from various sources, including molecular and morphological evidence. Common ancestors. A common ancestor is the most recent species from which two or more species have evolved. The branching points in a phylogenetic tree represent these common ancestors. 6. Importance of biodiversity and classification. Ecosystem stability. Biodiversity plays a crucial role in maintaining ecosystem services, such as pollination, soil formation, and nutrient cycling. Conservation efforts. Proper classification and understanding of biodiversity are essential for conservation strategies aimed at protecting endangered species and ecosystems. Medical and industrial applications. Many pharmaceutical drugs and industrial products are derived from naturally occurring organisms, highlighting the value of biodiversity. Previous year gave questions on diversity and classification of life. 1. Gate 2020 Question 1. Which of the following taxonomic ranks is the most inclusive? A. Family. B. Order. C. Kingdom. D. Species. Answer. C. Kingdom. Explanation. The kingdom is the most inclusive taxonomic rank, with several orders, families, and species included within it. 2. Gate 2017 Question 2. Which of the following is not a characteristic feature of archaea? A. Lack of a membrane-bound nucleus. B. Presence of peptidoglycan in their cell walls. C. Presence of histone proteins in their DNA. D. Ability to survive in extreme environments. Answer. B. Presence of peptidoglycan in their cell walls. Explanation. Archaea do not have peptidoglycan in their cell walls. This is a feature of bacteria. Instead, archaea have distinct cell wall structures. 3. Gate 2015 Question 3. The classification of organisms into domains, as proposed by Carl Woese, is based on A. Morphological characteristics. B. Genetic sequences, particularly ribosomal RNA. C. Fossil records. D. Biochemical similarities. Answer. B. Genetic sequences, particularly ribosomal RNA. Explanation. The classification into domains bacteria, archaea, and eukarya is based primarily on differences in the sequences of ribosomal RNA, rRNA, which reflect evolutionary relationships. Conclusion and application. The study of diversity and classification of life is fundamental to understanding how living organisms are related, how they evolve, and how they function in ecosystems. Through taxonomy, we categorize life into various hierarchical levels, with species being the basic unit. The advent of modern techniques, including genetic analysis, has revolutionized how we classify life and understand the evolutionary processes that lead to the rich diversity of organisms. For GATE, focusing on the hierarchical classification system, important taxa, and evolutionary relationships between organisms is crucial. Understanding the phylogenetic trees and the methods of classification, such as cladistics and molecular classification, will help you answer questions related to biodiversity, taxonomy, and evolutionary biology. You said, systems of classification. ChatGPT said, ChatGPT. Systems of classification. Classification is the process of grouping organisms based on shared characteristics. Over time, biologists have developed different systems of classification to categorize the diversity of life in a meaningful way. The goal is to represent evolutionary relationships and similarities among organisms. 1. Historical overview of classification systems. Historically, the classification of life forms was based on observable traits such as morphology and behavior. Over time, more sophisticated approaches, including genetic and molecular data, were incorporated. Some of the key classification systems in biological history include Linnaean classification, developed by Carl Linnaeus in the 18th century, this system uses a hierarchical structure and is based primarily on morphological features. The organisms are grouped into 7 primary taxonomic ranks, namely, kingdom, phylum, class, order, family, genus, and species. This system still forms the foundation of modern taxonomy. Phylogenetic classification cladistics. As our understanding of evolutionary relationships grew, classification systems were restructured to reflect the evolutionary history phylogeny of organisms. The goal is to group organisms based on shared ancestry rather than just physical traits. Cladistics became the most widely used method, focusing on the evolutionary lineage of organisms. 2. Taxonomic systems. The Linnaean system, traditional taxonomy. The Linnaean system is a hierarchical classification system that categorizes organisms into a series of nested groups based on shared characteristics. The primary levels, taxa, are domain, the highest level of classification, representing the most basic division of life. There are 3 domains. Bacteria, prokaryotic organisms without a nucleus, including common bacteria. Archaea, prokaryotic organisms that often live in extreme environments, such as thermophiles and halophils. Eukarya, organisms with eukaryotic cells, which include animals, plants, fungi, and protists. Kingdom, a large taxonomic rank below the domain. Some examples. Animalia, animals, plants, fungi, protista, protists. Formerly, prokaryotes, now split into bacteria and archaea. Phylum, a group of organisms that share a common body plan or structure. For example, Chordata, phylum that includes all animals with a notochord, vertebrates like humans, birds, etc. Arthropoda, phylum that includes insects, spiders, crustaceans, etc. Class, a subdivision of a phylum. For example, Mammalia, class that includes all mammals, characterized by the presence of hair and mammary glands. Aves, class that includes birds, characterized by feathers and beaks. Order, a division within a class. For example, Carnivora, order that includes carnivorous animals like cats, dogs, bears, etc. Family, a more specific group within an order. For example, Felidae, the family of cats, including lions, tigers, and domestic cats. Genus, a group of species that are closely related. For example, Panthera, genus that includes lions, tigers, and leopards. Species, the most specific level of classification, referring to a group of organisms that can interbreed and produce fertile offspring. For example, Homo sapiens, the scientific name for humans. The system of Linnaean classification is still in use, especially in traditional biology and botany. B. Phylogenetic cladistic classification. Cladistics is a modern method of classification based on evolutionary relationships phylogeny among organisms. The primary idea of cladistics is that organisms are grouped based on common ancestors and evolutionary traits. The system classifies organisms into clades, groups that include an ancestor and all its descendants. Key points include cladogram, a diagram that shows the evolutionary relationships between species based on shared derived characteristics, synapomorphies. Each branch point node represents the most recent common ancestor of the organisms at the tips of the branches. Monophyly, a group of organisms that includes a common ancestor and all its descendants. Monophyletic groups are preferred in cladistics. Synapomorphy, a shared, derived characteristic that is used to define a clade. For example, the presence of feathers in birds is a synapomorphy that groups them together. Paraphyly, a group that contains a common ancestor but not all its descendants. Paraphyletic groups are generally not preferred in modern classification. Cladogram, a diagram that shows the evolutionary relationships between species based on shared derived characteristics, synapomorphies. Each branch point node represents the most recent common ancestor of the organisms at the tips of the branches. Monophyly, a group of organisms that includes a common ancestor and all its descendants. Monophyletic groups are preferred in cladistics. Synapomorphy, a shared, derived characteristic that is used to define a clade. For example, the presence of feathers in birds is a synapomorphy that groups them together. Paraphyly, a group that contains a common ancestor but not all its descendants. Paraphyletic groups are generally not preferred in modern classification. Polyphyly, a group that does not include the most recent common ancestor of all its members. Polyphyletic groups are also avoided in modern phylogenetic classification. See molecular classification. Molecular classification is a modern method of classifying organisms based on molecular data, particularly DNA and RNA sequences, and protein sequences. This method provides insights into evolutionary relationships that may not be apparent from morphological characteristics. DNA sequencing. By comparing the DNA sequences of different organisms, scientists can construct phylogenetic trees and determine how closely related they are. Molecular clocks. The concept of using genetic mutations as a clock to estimate the time of divergence between species. Molecular classification has become essential in understanding evolutionary relationships at a more detailed level and has led to significant revisions in the traditional classification systems. 3. Modern classification systems. A 5-kingdom classification. Whittaker. In the 1960s, Robert Whittaker proposed a 5-kingdom system of classification based on cellular organization and modes of nutrition. Monera, prokaryotes, including bacteria and archaea. Protista, simple eukaryotes, including protozoa and algae. Fungi, non-photosynthetic organisms that absorb nutrients, e.g., molds, yeasts, mushrooms. Plantae, multicellular, photosynthetic organisms, e.g., mosses, ferns, conifers, and flowering plants. Animalia, multicellular, heterotrophic organisms, animals. B3 domain system. WOS. Carl Woese proposed a system in the 1990s that classified life into 3 domains based on genetic data, specifically ribosomal RNA, rRNA sequences. Bacteria, prokaryotic organisms with no nucleus. Archaea, prokaryotic organisms with distinct biochemistry and genetics, often found in extreme environments. Eukarya, organisms with eukaryotic cells, which include animals, plants, fungi, and protists. This 3-domain system, based on molecular evidence, has largely replaced the 5-kingdom system. 4. Importance of classification systems. Understanding evolution. Classification systems help us understand the evolutionary relationships between organisms, tracing the lineage of species back to their common ancestors. Identification and discovery. Classification allows scientists to identify, name, and categorize new species, which is crucial in biodiversity studies and conservation efforts. Communication. It provides a common language for biologists worldwide, making it easier to describe and study organisms. Conservation. Knowing the taxonomic status of organisms is critical for conservation, as it helps in identifying endangered species and monitoring biodiversity. Previous year gave questions on systems of classification. 1. GATE 2020 Question 1. In the phylogenetic classification system, the grouping of organisms is based on A. The size and color of organisms. B. Their evolutionary relationships and common ancestors. C. The number of chromosomes in an organism. D. The body structure of organisms. Answer. B. Their evolutionary relationships and common ancestors. Explanation. The phylogenetic classification system groups organisms based on their evolutionary history and shared ancestry. 2. GATE 2019 Question 2. In the Linnaean system of classification, the highest taxonomic rank is A. Kingdom. B. Phylum. C. Domain. D. Species. Answer. C. Domain. Explanation. The domain is the highest rank in the Linnaean system, above kingdom, and includes all life forms classified into 3 major domains: bacteria, archaea, and eukarya. 3. GATE 2018 Question 3. Which of the following classification systems is based on genetic data rather than morphological traits? A. Linnaean classification. B. Phylogenetic classification. C. Morphological classification. D. 5 kingdom system. Answer. B. Phylogenetic classification. Explanation. Phylogenetic classification is based on genetic data, particularly molecular sequences, to understand the evolutionary relationships between organisms. Conclusion. Classification systems provide a way to organize the vast diversity of life on Earth into manageable categories. The development from the traditional Linnaean system to more modern approaches, such as phylogenetic classification and molecular classification, reflects advances in our understanding of evolution and genetics. For GATE, it's essential to understand the principles of these classification systems, their evolutionary significance, and their application in modern biology. For GATE, it's essential to understand the principles of these classification systems, their evolutionary significance, and their application in modern biology. You said, Cladistics. ChatGPT said, ChatGPT Cladistics. Cladistics is a method of classification that organizes organisms based on shared evolutionary history, emphasizing their common ancestry. It is an essential approach in modern taxonomy and evolutionary biology, with a strong focus on grouping organisms by the most recent common ancestor, MRCA, rather than just morphological similarities. Cladistics allows for a more objective, scientifically grounded method of classifying life forms based on their evolutionary relationships. 1. Core concepts of cladistics. A clade. A clade is a group of organisms that includes a common ancestor and all its descendants. The key feature of a clade is that it represents an evolutionary lineage, showing how organisms are related through descent from a common ancestor. Clades can be large, such as the clade of all mammals, or small, such as the clade of all species within a specific genus. B. Synapomorphy. A synapomorphy is a shared, derived characteristic or trait that is present in an ancestral species and its descendants, distinguishing them from other organisms. These traits are used to define clades. For example, the presence of feathers is a synapomorphy for the clade containing birds and their closest extinct relatives, distinguishing them from other vertebrates. See phylogenetic tree. Cladogram. A phylogenetic tree, or cladogram, is a diagram that represents the evolutionary relationships between organisms. It visually shows the branching patterns of evolution and indicates how different species are related through common ancestry. The branches of a cladogram represent evolutionary divergences, while the nodes, branch points, represent the most recent common ancestor of the taxa involved. D. Outgroup. An outgroup is a species or group of species that is used as a reference to help determine the evolutionary relationships of the in-group, the group of species under study. The outgroup is assumed to be less closely related to the in-group, providing a comparative basis for identifying ancestral and derived characteristics. 2. Principles of cladistics. Cladistics is governed by several core principles that guide the construction of cladograms. An outgroup is a species or group of species that is used as a reference to help determine the evolutionary relationships of the in-group, the group of species under study. The outgroup is assumed to be less closely related to the in-group, providing a comparative basis for identifying ancestral and derived characteristics. 2. Principles of cladistics. Cladistics is governed by several core principles that guide the construction of cladograms. A monophyly. A monophyletic group is a group of organisms that includes a common ancestor and all of its descendants. A monophyletic group is considered a true clade. The goal of cladistics is to identify monophyletic groups that represent evolutionary relationships. Example, Mammalia mammals is a monophyletic group that includes all mammals and their most recent common ancestor. B. Paraphyly. A paraphyletic group includes a common ancestor but only some of its descendants. Paraphyletic groups are not considered clades in cladistics because they fail to represent the entire evolutionary lineage. Example, Reptilia reptiles, if defined to include only snakes, lizards, and turtles, but excluding birds, is a paraphyletic group because birds share a common ancestor with reptiles. See polyphyly. A polyphyletic group is a group that does not include the most recent common ancestor of all its members. Polyphyletic groups are also not considered clades, as they arise from multiple evolutionary lineages. Example, flying vertebrates, birds and bats, is a polyphyletic group because flight evolved independently in both lineages. 3. Construction of cladograms. A character selection. To construct a cladogram, scientists first select a set of characteristics or characters to examine in different species. These characters can be morphological, e.g., shape, size, molecular, e.g., DNA or protein sequences, or behavioral, e.g., mating behaviors. The characters must be carefully chosen to ensure that they reflect evolutionary relationships rather than superficial similarities. B. Character states. Each character is examined for its states or variations. For example, in the case of the character, presence of wings, the character states could be wings present or wings absent. The goal is to identify which character states are shared among the organisms being studied. See building the cladogram. Once the characters and their states are determined, the cladogram is constructed by determining which species share derived new character states that suggest they share a common ancestor. The more shared derived characteristics a group has, the more closely related its members are. Shared derived character synapomorphy. When two species share a derived trait that was inherited from a common ancestor, it is considered evidence of their evolutionary relationship. D. Parsimony principle. The principle of parsimony suggests that the simplest explanation, or the cladogram with the fewest evolutionary changes or character state changes, is preferred. This principle helps to avoid overcomplicating the tree and assumes that evolution tends to follow the least number of steps. 4. Cladistics versus traditional taxonomy. While traditional taxonomy, Linnaean classification, groups organisms based on observable similarities, cladistics groups organisms based on their evolutionary relationships. Some key differences include: taxonomy may group organisms with similar characteristics, morphology, without regard to their evolutionary relationships. For example, birds and bats were once grouped together as flying vertebrates, even though their flight is due to separate evolutionary events. Cladistics, on the other hand, groups organisms based on shared ancestry. For example, birds and dinosaurs, including extinct ones, are grouped together in a clade, despite differences in flight. 5. Applications of cladistics. Evolutionary biology. Cladistics is fundamental for understanding the evolutionary history of life on Earth. By constructing cladograms, scientists can trace the lineage of organisms, study how traits evolve, and understand the diversification of life. Conservation biology. Cladistics helps in identifying evolutionary distinct species that are important for conservation efforts. Protecting species that represent unique branches in the tree of life can preserve evolutionary history. Paleontology. In paleontology, cladistics is used to study extinct species and their relationships to modern organisms. Fossils are compared to living species based on shared traits to understand evolutionary transitions. Molecular phylogenetics. Cladistics is increasingly used in molecular phylogenetics, where DNA sequences and molecular data are used to construct cladograms and determine evolutionary relationships. 6. Advantages and disadvantages of cladistics. Advantages. Evolutionary insight. Cladistics provides a clear picture of evolutionary relationships based on shared ancestry. Objectivity. By relying on derived characters, synapomorphies, cladistics offers a more objective way to classify organisms compared to traditional, subjective methods based on superficial traits. Scientific rigor. The use of the parsimony principle ensures that the simplest and most efficient explanation for the data is selected. Disadvantages. Complexity. Constructing a cladogram can be complex, especially with large datasets and ambiguous traits. It requires detailed data and careful interpretation. Incomplete data. Incomplete fossil records or missing molecular data can complicate the reconstruction of evolutionary relationships. 7. Previous year GATE questions on cladistics. 1. GATE 2021 Question 1. Cladistics is a classification system based on A. Morphological traits. B. Evolutionary relationships and shared ancestry. C. Ecological roles. D. Nutritional strategies. Answer. B. Evolutionary relationships and shared ancestry. Explanation. Cladistics classifies organisms based on their evolutionary relationships, using shared derived characteristics. 2. GATE 2019 Question 2. In a cladogram, a node represents A. Common ancestor. B. A shared character. C. A branch point in the evolutionary tree. D. Both A and C. Answer. D. Both A and C. Explanation. A node at a point where evolutionary divergence occurs. 3. GATE 2018 Question 3. Which of the following best defines a clade? Four. Cladistics versus traditional taxonomy. While traditional taxonomy, Linnaean classification, groups organisms based on observable similarities, cladistics groups organisms based on their evolutionary relationships. Some key differences include: taxonomy may group organisms with similar characteristics, morphology, without regard to their evolutionary relationships. For example, birds and bats were once grouped together as flying vertebrates, even though their flight is due to separate evolutionary events. Cladistics, on the other hand, groups organisms based on shared ancestry. For example, birds and dinosaurs, including extinct ones, are grouped together in a clade, despite differences in flight. 5. Applications of cladistics. Evolutionary biology. Cladistics is fundamental for understanding the evolutionary history of life on Earth. By constructing cladograms, scientists can trace the lineage of organisms, study how traits evolve, and understand the diversification of life. Conservation biology. Cladistics helps in identifying evolutionary distinct species that are important for conservation efforts. Protecting species that represent unique branches in the tree of life can preserve evolutionary history. Paleontology. In paleontology, cladistics is used to study extinct species and their relationships to modern organisms. Fossils are compared to living species based on shared traits to understand evolutionary transitions. Molecular phylogenetics. Cladistics is increasingly used in molecular phylogenetics, where DNA sequences and molecular data are used to construct cladograms and determine evolutionary relationships. 6. Advantages and disadvantages of cladistics. Advantages. Evolutionary insight. Cladistics provides a clear picture of evolutionary relationships based on shared ancestry. Objectivity. By relying on derived characters, synapomorphies, cladistics offers a more objective way to classify organisms compared to traditional, subjective methods based on superficial traits. Scientific rigor. The use of the parsimony principle ensures that the simplest and most efficient explanation for the data is selected. Disadvantages. Complexity. Constructing a cladogram can be complex, especially with large datasets and ambiguous traits. It requires detailed data and careful interpretation. Incomplete data. Incomplete fossil records or missing molecular data can complicate the reconstruction of evolutionary relationships. 7. Previous year GATE questions on cladistics. 1. GATE 2021 Question 1. Cladistics is a classification system based on A. Morphological traits. B. Evolutionary relationships and shared ancestry. C. Ecological roles. D. Nutritional strategies. Answer. B. Evolutionary relationships and shared ancestry. Explanation. Cladistics classifies organisms based on their evolutionary relationships, using shared derived characteristics. 2. GATE 2019 Question 2. In a cladogram, a node represents A common ancestor. B a shared character. C a branch point in the evolutionary tree. D both A and C. Answer D both A and C. Explanation. Explanation. A node in a cladogram represents both a common ancestor and a branching point where evolutionary divergence occurs. 3. GATE 2018 Question 3. Which of the following best defines a clade? A group of organisms that share a common habitat. B a group of organisms that share a common ancestor and all its descendants. C a group of organisms with similar morphologies. D a group of organisms from the same genus. Answer B a group of organisms that share a common ancestor and all its descendants. Explanation. A clade is defined by shared ancestry and includes all descendants of the common ancestor, representing an evolutionary branch. Conclusion. Cladistics has become an indispensable tool in understanding the evolutionary relationships among organisms. By focusing on shared ancestry and derived traits, cladistics allows for a more accurate and evolutionary perspective on the classification of life forms. This method has significantly advanced the study of phylogenetics, offering insights into the deep evolutionary history of life on Earth. For GATE preparation, focusing on the principles, methods, and application of cladistics is essential. You said, and phonetics. ChatGPT said. ChatGPT phonetics. Phonetics, also known as numerical taxonomy, is an approach to classifying organisms based on their overall similarity or phenotypic characteristics. Unlike cladistics, which focuses on evolutionary relationships and shared ancestry, phonetics classifies organisms primarily based on observable traits, without considering evolutionary history. Phonetics uses a quantitative approach to assess the degree of similarity between different organisms, employing statistical methods to group them. Although it has been largely supplanted by cladistics in evolutionary biology, phonetics still plays a role in some areas of taxonomy and classification. 1. Core concepts of phonetics of phenotypic similarity. Phonetics relies on observable characteristics, known as phenotypes, to classify organisms. These can include external traits such as morphology, coloration, size, shape, and other measurable features. The basic assumption of phonetics is that more similar organisms are more likely to be closely related, irrespective of their evolutionary relationships. Example, in phonetic classification, two species of birds with similar beak shapes and body sizes would be grouped together, even if they evolved separately from different ancestral species. B. Numerical taxonomy. Numerical taxonomy is the application of statistical techniques to classify organisms based on multiple traits. In phonetics, a large number of characteristics, often hundreds or thousands, are compared across species. The data is then processed using cluster analysis or factor analysis to generate a classification scheme. Cluster analysis, a method used to group organisms based on similarity indices, e.g., Jaccard or Euclidean distance. Multivariate techniques, tools like principal component analysis (PCA) or discriminant analysis (DA) are used to analyze complex datasets and derive patterns. See similarity coefficients. Phonetic classification is often based on the similarity coefficient or distance matrix, which quantifies the degree of similarity or dissimilarity between pairs of organisms. These coefficients are used to calculate how closely organisms resemble each other across all the selected characteristics. Jaccard index measures the similarity of sample sets by comparing shared characteristics to total characteristics. Euclidean distance quantifies the straight line distance between points in a multidimensional space, e.g., size, shape, etc. 2. Process of phonetic classification. A selection of traits. The first. Multivariate techniques, tools like principal component analysis (PCA) or discriminant analysis (DA) are used to analyze complex datasets and derive patterns. See similarity coefficients. Phonetic classification is often based on the similarity coefficient or distance matrix, which quantifies the degree of similarity or dissimilarity between pairs of organisms. Classification is often based on the similarity coefficient or distance matrix, which quantifies the degree of similarity or dissimilarity between pairs of organisms. These coefficients are used to calculate how closely organisms resemble each other across all the selected characteristics. Jaccard index measures the similarity of sample sets by comparing shared characteristics to total characteristics. Euclidean distance quantifies the straight line distance between points in a multidimensional space, e.g., size, shape, etc. 2. Process of phonetic classification. A selection of traits. The first step in phonetic classification is to choose the traits that will be used to compare organisms. These traits should be measurable and distinctive. Both morphological traits, e.g., size, shape, color, and biochemical traits, e.g., protein patterns, enzyme activity.
ശരിക്കും ഒരു കണക്കിന് അറിയാമല്ലേ ആ ടീച്ചറെ സമ്മതിക്കണില്ല. ആ കോഴ്സിന് ചേർത്ത് വളരെ നന്നായി. നമ്മൾ ആ ചെറുപ്രായത്തിലെ വളരെ അഡ്വാൻസ് ടോപ്പിക്കുകൾ പഠിച്ചായിരുന്നു. ഇതൊക്കെ ഇപ്പം നോക്കി ഇതിപ്പോൾ MSc ലെവലല്ലേ, ഇപ്പോഴാണ് അതൊക്കെ അതിൽ വരുന്നത്. നമ്മളെ അപ്പോഴേ അത് പഠിപ്പിച്ചു. അതിനിപ്പോൾ വേണമെങ്കിൽ ഭാവിയിൽ ആ ബുക്ക് എടുത്ത് വായിച്ച് നല്ല മനസ്സിലാവും എന്തെങ്കിലും ആവശ്യമുണ്ട്. അതാണ് ഓരോ ദീർഘവീക്ഷണമുള്ള ആൾക്കാരാണ് ആ ടീച്ചർ. ജയശ്രീ ടീച്ചർ, ശാന്തി ടീച്ചർക്കൊന്നും അത്ര അറിയുന്നില്ല നേരെ പഠിപ്പിക്കും. പക്ഷെ എന്നാലും ശ്രദ്ധിച്ചിരുന്നോണ്ട് പിടിയിട്ടിട്ടോ.
Both morphological traits, e.g., size, shape, color, and biochemical traits, e.g., protein patterns, enzyme activity, can be considered. Example, for plants, traits like leaf shape, stem structure. Example, for plants, traits like leaf shape, stem structure, flower type, and color could be selected. B. Data collection. Data for the selected traits are then collected for the organisms being studied. This can involve direct observation or laboratory analysis, e.g., DNA sequencing, biochemical assays. In phonetics, more weight is placed on the number of traits rather than their evolutionary significance. C. Constructing a distance matrix. A distance matrix is constructed by calculating the similarity or dissimilarity between pairs of organisms based on the traits being analyzed. This matrix allows the clustering of organisms based on their overall similarity, forming groups clusters of organisms that are phenotypically similar. D. Cluster analysis. Using the distance matrix, organisms are grouped into clusters that represent phenetic relationships. These clusters are then arranged into a dendrogram, a tree-like diagram that shows how closely related different species are based on their traits. E. Classifying organisms. The final step is the classification of organisms based on the clusters obtained from the analysis. The resulting classification is often a phenetic taxonomy, which groups organisms based on their phenotypic similarity, regardless of their evolutionary history. 3. Phonetics versus cladistics. Phonetics and cladistics are both methods of classification, but they differ significantly in their approach and goals. A. Phonetics focuses on overall phenotypic similarity, using observable traits, does not consider evolutionary relationships or common ancestry, employs statistical and mathematical methods to quantify similarity, groups organisms by overall resemblance, not by shared evolutionary history. B. Cladistics focuses on shared ancestry and evolutionary relationships, uses synapomorphies, shared derived traits, to classify organisms, groups organisms based on the most recent common ancestor, MRCA, aims to create monophyletic groups that represent true evolutionary lineages. Comparison. While phonetics is based on trait similarity, cladistics groups organisms by evolutionary history. Cladistics is considered more scientifically rigorous for understanding evolutionary relationships, whereas phonetics is more straightforward and can be useful for initial, large-scale classification. 4. Applications of phonetics, a large-scale taxonomy. Phonetics can be useful for the initial stages of classification, especially when working with large datasets or when evolutionary relationships are not well understood. It is often used for grouping organisms with many shared traits that do not yet have well-defined evolutionary histories. B. Species identification. Phonetic methods can be employed in species identification, especially in fields like forensic biology, biodiversity surveys, and botany, where quick and straightforward methods of classifying organisms are needed. C. Fossil classification. In paleontology, phonetic classification can be applied when fossil organisms are poorly understood or lack clear evolutionary relationships. The use of observable characteristics allows researchers to compare extinct species with living relatives. 5. Criticisms of phonetics, a lack of evolutionary context. One of the main criticisms of phonetics is its disregard for evolutionary relationships. Organisms that are phenotypically similar but not evolutionarily related can be grouped together, which may lead to incorrect conclusions about their evolutionary history. B. Oversimplification. Phonetics focuses primarily on trait similarities, which can oversimplify the complexity of evolutionary processes. Traits that are convergent evolved independently may be treated as homologous, which can lead to misleading classifications. C. Lack of focus on derived traits. Phonetics does not differentiate between derived and ancestral traits, which are crucial in understanding evolutionary relationships. Cladistics, by contrast, focuses on synapomorphies, which are derived traits that indicate a closer evolutionary relationship. 6. Previous year GATE questions on phonetics. 1. GATE 2020 Question 1. Which of the following is a key characteristic of phonetic classification? A. It focuses on evolutionary relationships based on shared ancestry. B. It groups organisms based on observable phenotypic similarity. C. It uses only molecular data for classification. D. It does not consider phenotypic traits. Answer. B. It groups organisms based on observable phenotypic similarity. Explanation. Phonetics classifies organisms based on their overall phenotypic traits, such as morphology and biochemistry, without considering their evolutionary history. 2. GATE 2017 Question 2. In phonetic classification, the method that uses statistical techniques to group organisms based on their traits is known as A. Cladistics. B. Phonetic distance analysis. C. Molecular phylogenetics. D. Evolutionary taxonomy. Answer. B. Phonetic distance analysis. Explanation. Phonetic classification involves the use of distance analysis to quantify similarity and group organisms based on phenotypic traits, often employing methods like cluster analysis. 3. GATE 2015 Question 3. Which of the following is not a common technique used in phonetics for classification? A. Cluster analysis. B. Principal component analysis. C. Synapomorphy analysis. D. Factor analysis. Answer. C. Synapomorphy analysis. Explanation. Synapomorphy analysis is a key feature of cladistics, not phonetics, as it focuses on shared derived traits to define evolutionary relationships. Conclusion. Phonetics, though largely replaced by cladistics in modern evolutionary biology, remains a significant tool in the classification of organisms based on phenotypic similarity. It is particularly useful for large-scale classifications or when evolutionary relationships are unclear. Understanding phonetics, their applications, and limitations is important for a comprehensive study of taxonomy and evolutionary biology, especially for GATE preparation where knowledge of both phonetics and cladistics is crucial. You said. Applications of both these. ChatGPT said. ChatGPT. Applications of phonetics and cladistics in evolutionary biology. Both phonetics and cladistics have significant applications in the study of biodiversity, taxonomy, and evolutionary biology, but they are used in different contexts based on their underlying principles and objectives. 1. Applications of phonetics. Phonetics is primarily concerned with grouping organisms based on their overall phenotypic similarity, without considering their evolutionary relationships. This approach has several important applications, especially in situations where evolutionary relationships are either unclear or difficult to determine. A. Taxonomy and classification. Initial grouping of organisms. Phonetics is widely used for grouping organisms based on observable traits, e.g., morphology, size, shape, and other phenotypic features. This method is useful when scientists need to classify large groups of organisms or when evolutionary relationships are not well understood. Biodiversity surveys. In biodiversity studies, phonetics can be employed to quickly group organisms in a given area based on shared traits, especially in situations where molecular or genetic data may not be available. Morphological classification. For organisms with limited molecular data, e.g., extinct species or fossilized organisms, phonetics offers an alternative by focusing on morphological traits to assign them to higher taxonomic groups. B. Fossil classification. Fossil identification. Phonetics is commonly used in paleontology to classify and identify fossil organisms, particularly when molecular data is not available. The phenotypic features of fossils, e.g., skeletal structure, size, shape, are used to compare them to living organisms. Reconstructing evolutionary history. Although phonetics does not focus on evolutionary history, it can provide valuable clues about the diversity of life forms in the past and help identify broad trends in the evolution of species. See forensic biology. Species identification. In forensic biology, phonetic methods are often applied to identify species based on their physical characteristics. For example, in criminal investigations involving animal remains, phonetic classification can help match samples to known species based on phenotypic traits. Tracking biodiversity. In conservation biology, phonetic methods can assist in monitoring species diversity in ecosystems, providing quick and reliable classifications to track the health of species populations. D. Ecological studies. Functional traits analysis. In some ecological studies, phonetics is used to classify species based on functional traits like leaf morphology or flower shape. This can help understand ecological roles, such as pollination mechanisms or habitat preferences, without needing detailed genetic data. 2. Applications of cladistics. Cladistics, which focuses on grouping organisms based on shared ancestry and evolutionary history, is the cornerstone of modern evolutionary biology. Its applications are primarily centered around understanding the evolutionary relationships between organisms. Understanding evolutionary relationships. Phylogenetic tree construction. One of the most significant applications of cladistics is the construction of phylogenetic trees that depict the evolutionary relationships among species. These trees are built using shared derived traits, synapomorphies, and represent the branching patterns of common ancestry. Evolution of traits. Cladistics helps in tracing the evolution of specific traits, e.g., the evolution of wings in birds or the development of flowers in plants by identifying when and how these traits arose in evolutionary history. B. Systematics and classification. Creation of monophyletic groups. Cladistics aims to classify organisms into monophyletic groups, which are groups that contain all descendants of a common ancestor. This approach ensures that the classification reflects true evolutionary relationships rather than just superficial similarities. Revising taxonomy. By applying cladistic methods, scientists can reevaluate traditional taxonomic classifications that were based on phonetic traits and may have been misleading. For example, species that were previously classified together based on similarity may be placed in different clades if their evolutionary histories suggest otherwise. See evolutionary biology. Studying evolutionary divergence. Cladistics plays a vital role in understanding how different species diverge over time. By constructing phylogenies, scientists can infer how certain species adapted to specific environmental pressures and developed unique traits over evolutionary time. Examining adaptive radiation. Cladistics can help trace the phenomenon of adaptive radiation, where a single ancestral species diversifies into many different forms. By analyzing the evolutionary branches of species in a clade, researchers can understand how adaptive radiation has led to biodiversity. D. Conservation biology. Identifying evolutionarily significant units, ESUs. In conservation biology, cladistics can be applied to identify evolutionarily significant units, ESUs, which are populations of species that have a unique evolutionary history and are important for conservation efforts. This ensures that conservation priorities are based on preserving evolutionary lineages rather than just protecting superficially similar species. Genetic conservation. In cases where species are threatened with extinction, cladistics is used to ensure that genetically distinct populations are preserved. This is important for maintaining genetic diversity within species and ensuring long-term survival. E. Molecular evolution. Molecular phylogenetics. Molecular data, e.g., DNA or protein sequences, is increasingly being used in cladistics to build phylogenetic trees. This method helps to clarify evolutionary relationships at the molecular level and resolve disputes in taxonomy based on phenotypic data alone. Gene evolution. Cladistics can be applied to study the evolution of genes and genomes, tracing how specific genes or gene families evolved across different species. This is particularly useful in understanding evolutionary processes at the molecular level. F. Paleontology. Fossil reconstruction. While phonetics is often used for fossil identification, cladistics plays a role in reconstructing the evolutionary history of extinct species. By comparing fossilized remains with living relatives, cladistics can help determine the evolutionary lineage and ancestral traits of extinct species. Tracking evolutionary trends. Cladistics is also used in paleontology to trace major evolutionary trends over time, such as the development of new body plans, adaptations to changing environments, and the evolution of key traits in the fossil record. 3. Comparison of applications in different contexts. Application phonetics cladistics. Taxonomy initial grouping of organisms based on overall similarity, e.g., morphological traits classification based on shared ancestry and evolutionary relationships. Fossil classification used when molecular data is unavailable. Relies on phenotypic traits for classification. Helps reconstruct evolutionary history of extinct species using shared derived traits. Biodiversity studies classifies organisms quickly based on phenotypic traits. Identifies evolutionary lineages and relationships between species. Conservation biology helps classify species or groups quickly for monitoring. And management focuses on preserving evolutionary lineages, including evolutionarily significant units, ESUs. Evolutionary studies focuses on observable trait similarity, often without regard to evolutionary relationships. Aims to understand evolutionary divergence and adaptive radiation using phylogenetic trees. Genetic studies rarely used in molecular genetics. Essential in studying molecular evolution and tracing gene family evolution. Ecological studies used for grouping organisms based on functional traits in ecosystems. Focuses on evolutionary adaptations to ecological niches. And environments. Conclusion. Both phonetics and cladistics are valuable tools in evolutionary biology, but their applications differ significantly. Phonetics is best suited for classifying organisms based on observable characteristics, especially when evolutionary histories are not well known, while cladistics excels in understanding the evolutionary relationships and history of organisms. As both methods have distinct strengths, they can complement each other in certain contexts, such as early-stage classification. Phonetics followed by a more detailed analysis of evolutionary relationships. Cladistics. Understanding the proper application of each approach is essential for a well-rounded understanding of biological classification and evolutionary processes. You said. Life history strategies. ChatGPT said. ChatGPT. Life history strategies. Life history strategies refer to the patterns of growth, reproduction, and survival that organisms exhibit over their lifetime. These strategies are shaped by evolutionary pressures and trade-offs that organisms face in response to their environments. Life history theory seeks to explain how natural selection influences the allocation of energy to different life stages and the types of reproductive strategies that maximize an organism's fitness in a given environment. 1. Key concepts in life history theory. Life history strategies are broadly categorized based on how organisms allocate their resources, e.g., energy, time, to different life functions such as growth, reproduction, and survival. These strategies are often viewed as existing along a continuum, with organisms exhibiting various combinations of characteristics. Trade-offs. The primary concept in life history strategies is the trade-off between different life functions, especially reproduction and survival. Because resources are limited, an organism must allocate them in a way that maximizes its reproductive success while ensuring survival. Examples of trade-offs. Quantity versus quality of offspring. Organisms can either produce many offspring with less investment in each R strategy, or fewer offspring with more parental investment K strategy. Growth versus reproduction. Species must decide whether to allocate resources toward faster growth or earlier reproduction, which can have consequences on lifespan and fitness. B. Fitness maximization. The central aim of life history strat.